Microscope class part 2, Electron Microscope. In this session, we will discuss about the electron microscope such as scanning electron microscope and transmission electron microscope and their working principle and functions. Electron microscope. In 1934, Ernest Rucker developed the electron microscope. By this electron microscope, very tiny particles, or mainly the virus, can be observed through this electron microscope. Because viruses are very, very tiny organism, it is measured in nanometer when compared to the bacteria. The bacteria is measured in micron or micrometer. So, with the help of this simple or compound optical microscope, it is not possible to see the virus. Only with the electron microscope, we can observe the morphology of virus. In electron microscope, we use electron instead of light when comparing to the optical microscope. And this shorter wavelength of electron will give greater resolution. So, you will get a better or clear image of the microorganism. And there are two types of electron microscope are there. First one is a scanning electron microscope, and the second one is the transmission electron microscope. Scanning electron microscope, it is also called as SEM. Scanning electron microscopy obtain high resolution image and detailed surface information of the sample. The scanning electron microscope uses a focused beam of electron to scan the surface of specimen and generate greater resolution of image when compared to the optical microscopy. Simply, we can say that in scanning electron microscope, this microscope will scan the image or scan the specimen. So, we will get a better or high quality, greater resolution of image can be observed. And this is the picture of the scanning electron microscope. And the scanning electron microscope is connected to the computer monitor with the software. So what and all we are observing inside this scanning electron microscope can be visualized in the computer monitor. Scanning electron microscope. Now we will discuss about the working principle of scanning electron microscope. First, about the part of scanning, different parts of scanning electron microscope. This is the electron source. This is the anode. This is a condenser. This is the objective lens. This objective lens is connected with X and Y scan coil. This is connected to the computer. And these are the signal detector, X-ray detector, backscattered electron detector, secondary electron detector, and here we will keep the sample. The sample is kept on a mounting stage. And this is the electron source. This electron source consists of electron gun. Mainly tungsten filament are used as electron gun. This electron gun will produce electron. This electron will reaches to the condenser through this anode ring. The anode ring will give a voltage. It will accelerate this electron source or electron beam, and it will focus the electron is focused to the condenser lens. And this condenser lens will reduce the beam diameter of electron. And the objective lens will focus the beam on the sample surface. The position of the electron beam on the sample is controlled by scan coil above the objective lens. That means the coil allows the beams to scan over the surface of sample in X and Y plane. The sample is mounted on a stage in a chamber at vacuum. That means this area are in vacuum. When electron pass and hit the sample, it will produce or it will generate various type of electron signal. And these electron signal are detected by these detectors such as X-ray detector, backscattered electron detector, and secondary electron detector. And these are connected to a computer with a software. In simply, we can say that in a scanned electron microscope, the detector will detect the scattered light intensity. The difference in scattered light intensity will transfer to the computer. So, the computer will give a high magnification of the scattered area or sample in the chamber. So, in scanning electron microscope will provide the information on the surface of sample like roughness or contamination detection. That means in scanning electron microscope we scan the area and it will scatter the light or it will emit some electron that will be detected. So we will get an idea about the surface of the sample and the surrounding area. The sample surface and its composition by evaluating the scattering light detected by the coil. And its resolution limited 0.5 nanometer and it will provide 3D image of the surface. So here you can see this is the 3D image of the observation and no need any specific requirement for sample preparation. So you don't have to do any specific procedure for the sample preparation. Just we will take the sample and we will keep it in the sample chamber and we will observe under this microscope. And it accelerates voltage up to 330 kV, not 330 kV. The magnification limited up to 1 to 2 million times. And the maximum field can be covered with higher depth of image. And when comparing to this other electron microscope, this procedure is very simple to operate. And this is the microscopic image of nanofibers, or we can say this is a comparison image of nanofibers from compound microscope and the scanning electron microscope. By seeing these pictures, we will get an idea how the depth and resolution of this image quality of the scanning electron microscope. Transmission electron microscope, it is also called as TEM. In transmission electron microscope, use a beam of electron to create an image of the internal structure of thin sample such as molecule and tissue section. That means here we will observe the internal structure of thin sample of molecules and tissue sections. The electron beam will pass through the thin sample and creating a projection of image in the computer. Here you can see this is the transmission electron microscope column or chamber or tube, and after that the projected image can be observed in this computer. And in this transmission electron microscope will magnify nanometer structure up to 50 million times when comparing to the other microscope. The working principle of transmission electron microscope, and this is the tube of transmission electron microscope. This tube is in vacuum. That means there is no air is present inside this tube. And this tube is again divided into illumination system and image formation area and image magnifying area. So this is the electron source. This electrons from the electron source, electron will be produced. So this electron beam will reaches to the condenser. The condenser will narrow the diameter of this electron beams, and also this condenser and this objective lens will focus and project the electron beams to the sample. And the presence of this objective lens and the objective aperture will enhance the magnification of the transmitted image. And this is the intermediate lens. An intermediate lens will also enhance or enlarged image when these beams, transmitted beams, reaches to the projector lens. This projector lens will project the image into this fluorescent screen, and it is connected with the computer. So the projected image can be observed in the computer monitor. So in transmission electron microscope, the electron will transmit or pass through the sample. So the inner structure of the sample can be observed like crystal structure morphology. So with the help of this transmission electron microscope, we can clearly observe the inner structure of the smear or the sample or the cell or bacteria or virus, whatever it is. And the resolution is less than 50 picometer. 1 picometer is equal to 1000 nanometer. So you can imagine how much is the 50 picometer. And it provides 2-dimension projection in the sample. The sample preparation is complex when compared to the scanning electron microscope. And the sample must be thin, generally below 150 nanometer. It accelerates voltage range of 60 to 300 kV. It magnifies more than 50 million times. And a very small field or small part of the image can be seen through this transmission electron microscope when we are comparing with the scanning electron microscope. So this is the comparison of image produced or generated by the transmission electron microscope and scanned electron microscope. Scanned electron microscope, we will get a 3-dimensional picture of the sample with the surface. Not only this is sample, the surface can also be visualized by the help of this scanning electron microscopy. Here you can see in transmission electron microscope, the light or the electron beam will transmit or pass through the sample, so we can clearly see the internal structure of the specimen.
Subscribe to Simply Micromed. If you're curious about medical microbiology, infectious diseases, and unraveling the mysteries of tiny organisms, hit that subscribe button. Join our community of science enthusiasts, and let's explore the microscopic world together. Remember, knowledge is contagious. Spread it by liking and subscribing. Thank you.